Okay, so on to our second to last lecture of the semester, which is connecting genotypes to phenotypes, right? All right. So as we've talked about actually this past class, this past week, one of the interesting things about our genetic code is that different organisms or organisms that have different kinds of cells can share all their own genetic information, right? So again, human cells, for example, we have 46 chromosomes, and those same 46 chromosomes begin through, again, the process of uh, fertilization of sperm to egg, and then that creates a zygote. And that zygote will eventually grow into all of the different cells that we find in a human. Same thing for plants, right? Plants begin as sperm and egg, and after the zygote is formed, the zygote grows into a variety of different kinds of cells that have different kinds of functions. But again, the important thing for this it really is the fact that, when you have a certain genetic construct or a genotype, right? All of the genes that all of the cells in any particular organism have access to are confined within the genes that have been inherited from their parents. And of course, this also reigns true for prokaryotes and fungi and protists, but for single-celled organisms, right? So for single-celled organisms, their phenotypes are a little bit more complicated because they don't have truly different cell functions per se, but one cell compared to another cell might have slightly different biologies, right? They might have different metabolic rates, they might have different sugar requirements, they might be doing different things, they might be in different stages of their cell cycle or in their binary fission state. And so, not so surprisingly, all of those features and functions of what genes are being made into protein. So, what proteins are we making, and how much of this protein are we making? Um, are two really important kind of concepts to what actually is defined as our phenotypes. So, just to reiterate, right, when we think about the process by which this occurs in eukaryotes, every gene that is found in the DNA equates to one protein. And for that matter, every gene in any living thing equates to one protein. The question really becomes how many genes are made into protein per transcriptional unit. And we'll talk a little bit about this when we talk about transcription and translation. But for now, to understand what we're talking about, we know that in DNA, we have different forms of DNA, we have different sections of DNA, and DNA therefore has spaces, right? It has nucleotides. These nucleotides are going to be read uh, out as a message, and then that message is going to then dictate the sequence of amino acids that come off the ribosomes that come off the ribosome, and then proteins will fold according to their sequence of amino acids and their environment. So this leads us to our central dogma, where DNA houses all of the nucleic acid information required for maintaining a genetic code that is then transcribed into RNA, and the RNA is then sent to the ribosomes. And then in the ribosomes, this is where we get the process of translation, where we get an amino acid sequence, and the amino acid sequence folds into the tertiary or quaternary protein. So DNA has all the instructions you need to make all the proteins. And the important thing is when we think about genotype and phenotype, well, what is phenotype exactly? phenotype is the expressivity of a genotype. It has everything to do about the characteristics of an organism. So everything from what they look like, right? Do you have a long nose? Do you have blue eyes? Do you have uh, black hair? Versus, other things that we think about that are phenotypic, but maybe not as easy to kind of relate to as phenotypes is how well do you digest lipids? How is your metabolism of phospholipids? Do you make a lot of important molecules from your phospholipids? What blood type are you? Are you A, B, O? Do you have rheumatoid factor? Other phenotypes could be, uh, your even as simple as your blood pressure, or your resting heart rate. These are all things that are, or functions that are related to proteins or the biochemistry and the biosynthesis or other type of reactions of proteins. So proteins do all of the work of a cell. And because they do all the work of the cell, and proteins therefore are the phenotypic connection, right? We call our genes in all of our DNA our genotype. And therefore, all of the things that proteins do, right? The work they do, their function, is equated to their phenotype. And this is important because when you think about phenotypes, you might say, okay, well, if I'm looking at skin color, that is cells. And so the outer aspect of cells is cell membranes, and that's phospholipids. That's true. Or we might be looking at pigmentation. We're talking about eye color, not proteins. It's pigmentation. They're pigments. They're colors. And that's also true. But who makes the pigment? Who puts in the pigment? Who is responsible for the biosynthesis of the lipids in the membrane? They're all functions of the proteins. So when we think about this, right, again, the genetic information is housed in the nucleus of all cells, and then this is the site, right? The nucleus is the site of transcription. And for eukaryotes, transcription occurs inside the cell. The RNA is then shuttled out into the rough ER, where it then makes protein, right? And the thing is, all the different parts of a cell, even, right? How many mitochondria there are, what is the thickness of their cell wall, what is the um, comp composition of the cell membrane, right? What is the composition of the membranes of the organelles, the functions of the organelles within the cell, and therefore the functions of the cell themselves. Again, are all part and participle of the proteins that that cell can create. So when we think about this, again, we talk about mitosis, meiosis, vertical transfer or heredity of genetic information versus the outsourcing and evolution brought on by mutation and things like meiotic crossing over. Even though we talk about the movement of chromosomes, we talk about eukaryotes especially, what are we really moving? What are we inheriting? What is the molecule of inheritance? That is the chromosome, because inside the chromosome, if we go all the way down to what is housed inside of the fibrous chromosome, that's about 1,400 um, nanometers wide, we then eventually get down to individual nucleosides. Again, when we think about what is the part of the DNA that is variant, the part of the DNA that could have differences in its sequence or differences in the letters associated with the message that is going to define the phenotype, that is definitely in the nucleosides, because the backbone, right, the sugar phosphate backbone is invariant. So this is why the sequence and the sequence that is housed here, if DNA, right, if the sequence of DNA results in the sequence of RNA that then results in the sequence of amino acids that make the protein, It is the direct correlation of the genotype and the phenotype, which is also why things like mutations that we talked about in this class can be so devastating. Because if you change the sequence of these things, you inevitably could change the sequence of these things, and therefore the phenotype. So it's important to note, right, when we transcribe a gene, each individual gene, not necessarily mRNA, for eukaryotes, as we'll see later, for eukaryotes, one mRNA results in one protein. In prokaryotes and in viruses, one mRNA typically results in more than one protein. But even in these cases where we have one mRNA, one protein, or one mRNA, multiple proteins, what defines each individual protein? That is the dimensions or um, the edges of a gene. A gene has multiple important components to it that help define what it is. Genes have a place where RNA polymerase can make the RNA. This is called the promoter. The promoter is then flanked, uh, heading again, this is the 5' end, heading towards the 3' end. There is the coding sequence, and the coding sequence is what is going to actually make the protein. And so each individual gene, if you have one mRNA, one protein, you have one coding sequence because it'll have a defined 3' end or a stop and a start. And so you have a start and stop, and that whole thing is coded for. When you have multiple, it's the same structure. You have a promoter, but now instead of one coding sequence, you just have multiple coding sequences with multiple start and stop sequences, and then with a defined 3' end. So there are a lot of different kinds of proteins, and we've talked about these before, but just to kind of go back through these, right? There are digestive enzymes. These are enzymes that break down things, and a lot of these have the end ASE in them. Amylases are those that help break down food, lipase for lipids, pepsins or proteases for proteins, and then trypsin is a different kind of um, protein breaking apart enzyme. They're a transport. These can be heterogeneous, like hemoglobin. So these are ones that actually are moved by something else. So hemoglobin is found in blood, and blood moves that. Transport can also be on the cell membrane, right? We have carrier receptors and active transport, right? So it can either be something that carries something, down the bloodstream, which is carrying something, or transporting something just simply across a membrane. Structural, this is actin, tubulin, keratin. Tubulin we talked about. This is what makes up uh, the fibers that are used in mitosis. Whereas actin, myosin, and keratin make up other structural components. They play an important role in the cytoskeleton. Hormones, insulin, thyroxine, they help with the body and they help coordinate different actions. Hormones can also be found in prokaryotes and single-celled eukaryotes. So signaling molecules and signaling proteins, signaling peptides. Defense, again, you could have immunoglobulins, which are antibodies. So, you get a vaccine, get antibodies. Contractile, actin and myosin. This is found mostly in eukaryotes, but you can find at least actin in a lot of prokaryotes. And then storage. places where you can have proteins that have a non-catalytic substrate binding site, and they can store large molecules that can be then accessed for cell function after the fact. So in humans alone, there are roughly about 20,000 proteins in the human body. They all have different structures, they all do different things. But how can they do that? How are they can they diversify in such a way? Well, a lot of the fact is that the function of the function and location of a protein has to do with the amino acids that make it. So the sequence of the amino acids are going to ultimately define the protein shape. The protein shape is going to define several features of the protein, especially the amino acids that lie on the outer cortex or the outer ridge of the protein. And these are the amino acids that typically will allow the amino acid to exist somewhere in space. So, for example, if it's a membrane protein, if so going back, several lectures, we have integral proteins that are in the membrane. And then we also have non-integral proteins, right? We have accessory proteins that sit here in the membrane like so. So again, these are polar heads, so the amino acids in this phase of the protein must be also polar because that's how they interact. Whereas this protein might have polar and then non-polar amino acids at the ridging so that it sits both with the phosphate heads and within the lipid cortex of the membrane. The other aspects of the amino acids also determine its shape in whether or not it can bind to a target protein. So why receptors are specific to their target molecules, why enzymes are specific to their ligands, is because the amino acid folding isn't just about the body of the protein and its location, but also gives it the right shape or cleft along with the right chemistry or amino acids to do the chemistry required of that enzyme, whether it's to bind and relay phosphate information or whether it is to chemically modify or structurally modify a target molecule. So we do this because we have 20 different kinds of amino acids. They all have different R groups. All of the R groups that you see in different kinds of amino acids allow them to do different kinds of chemistry, allow them to make all these different folds and all these different shapes, and allow for mobilization or kind of reaction with the protein to whatever the job of the protein is. So proteins typically fall under one of four categories. There are the nonpolar amino acids. They are here, of which there are 10. All of these don't have any particular charge. So again, the R group is here. It's highlighted because again, the amino acid is defined by this sequence here. Sorry, this sequence here. So we have the amine group, the carboxylic acid group, that flank a central carbon, and off that central carbon is where you get the R group. And the R group defines the structure and or chemistry of that particular amino acid. So for example, not all nonpolar amino acids are created equal. You have some that are simple, so some that have these uh, different spacings of methyl groups. Some are linear, some are bifurcated, some have nitrogenous rings, others have just cyclic benzene rings. And again, these two, phenylalanine and tryptophan, have these giant ring structures attached to them. They are technically nonpolar, but they are a lot larger than all of these other amino acids. And then you have a really interesting amino acid that is nonpolar, which is proline. And proline has this pentameric structure that links to the actual amine group. So the R group actually cycles back to the amine group, creating this pentameric shape. And so you can see that there are different shapes here and there are different chemistries, right? For example, you have methionine, it has a sulfur that abridges two methyl groups, which is unusual. And all of these different things do play an important role in whether or not or how these different nonpolar amino acids interact. So it isn't just simply it is not just you cannot just simply say, well, an amino acid is nonpolar, so they all act the same. They all don't act the same because they all have different structures. And even though the chemistry is the same, because the charge and or access to different kinds of the access to certain kind of elements or atoms to do certain chemistries might be the same because they're all nonpolar, but the structures are where they vary in the structural component of the protein, which defines its function. There are also polar amino acids, um, of which there are five. These are all defined by the fact that they do not have a charge, but they have a partial charge. So they have typically most of them have an actual hydroxide group somewhere along their structure, or they have something like an amine group that has the ability to have a relatively uh, complex chemistry or that partial charge chemistry that is found in polar molecules. Again, some of them have rings like tyrosine. You also have threonine, which has this bifurcated structure, and then asparagine and glutamine having these um, amide groups at the end of their terminal carbon. So we have polar uncharged, we have nonpolar uncharged, and so the last two that we have are charged particles or charged amino acids. There are positively charged ones. There are only three: lysine, arginine, and histidine. Lysine, arginine, and histidine all have these positive charges at the end of their sequences or the end of their R groups. This allows them to create salt bridges, ionic bonds, and things of that nature. And then we have aspartic acid and glutamic acid, only two that are negatively charged. They're negatively charged because um, they swap out their amide groups for an oxygen. The oxygen is a -2, so with the one, they each have one valent electron. So the oxygen maintains a negative charge, which is important for it to be able to be hydrogenated or otherwise modified. So again, uh, the shape of a protein does determine its function, and that's why again the sequence and all the aspects of an amino of a protein are so important. So the sequence is really important. So what sequence we have and composition, right? We have different types of amino acids. So we have histidine, which is a positively charged amino acid. Valine, leucine, threonine. Um, these are all uh, non-polar. Then we have proline, which is non-polar, but as the pentamer. Now we have glutamine. Glutamine being again, these two are go back. Sorry. Glutamine is polar, right? So we have polar, nonpolar, and we have a positively charged amino acid. So again, the compositional heterogeneity we have in these particular amino acids defines the ability for this protein to fold correctly. And it's not just the sequence, again, right? It's also the environment, the pH, the oxygen content, the water concentration, all of these things, right? Temperature, all of these things play an important role in how these are going to interact with one another. So not so surprisingly, right? If we change the order or if we change the composition, so in this case, like sickle cell that you learned in lab, we're not changing the order, we're just changing the composition. We're changing out a glutamate, which is again which is a polar amino acid, to a valine, which is a nonpolar amino acid. And this causes uh, a hydrophobic linker. which creates a protein aggregate and prevents the actual proteins from functioning correctly. So again, it's really important that the sequence is correct. But how do we know how to get that sequence? That again is from the DNA, because the DNA holds the key to make our amino acid uh, from the RNA that is going to be read out as the message from the DNA. All right. So when we think about this, right, the sequence of the DNA is really important because then that defines the mRNA message. Now, the interesting thing is, as we've talked about, DNA comes in two strands. There is the 5 to 3 strand, and then there is the 3 to 5 strand. And as we've discussed, how the polymerases work is they read 3' to 5' because they're going to synthesize 5' to 3', right? So they're reading in this direction, but they're synthesizing in this direction, which means that in reality, this bottom strand here, this is our template strand. So this is going to be what tells us the order of nucleic acids are going to be for the actual instructional piece. And we know this because, as you might recall from DNA replication, right, we had two strands of DNA. If we're reading in, if we're going in the opposite direction, again, this is 3', this is 5', we need to be moving in this direction, right? We're synthesizing in 5' to 3', but how are we doing that? We're doing that with RNA primers, right? So again, RNA very transiently makes this heteroduplex with RNA and DNA, and that then will translate into the RNA that we're creating. So the top strand here, therefore, is what we call the coding strand. So we call it the coding strand because it's the sequence that we're going to read or translate into our RNA. So if the DNA says TTC on the coding strand, right, the mRNA is AAG. And again, because we're reading in the opposite direction, so we're doing it in complement. So TTT equals AAG. AAG is lysine. So again, this order of sequence is really important because then that defines the amino acid that we create. Now, mutations can come, it can be important in a couple different ways. Mutations can be important by way of evolution. So things like bacteria, fungi, protists, viruses, these types of organisms depend on point mutations in order for evolution to occur. For humans and for plants and animals, for multicellular eukaryotes, point mutations are a slow but definitive way of evolution. And we've talked at length about this, that our evolution is slow because we sexually reproduce. Therefore, the only chance by which we can have evolution through creation of progeny is through mutations that occur through meiotic recombination and somatic mutation. But as we've also discussed, there is also the propensity for the mutations to go wrong. And so when we have mutations that change the overall sequence, or they change the nucleotide instructions, the question then becomes, do they change the overall amino acid that is produced, or do they change something about the gene that we are trying to express? So mutations come in three different forms, and one of them actually has two different kinds of forms. So the first is called a silent mutation. So our sequence here is TTC. The mutation here is TTT. Now the cool thing is the mRNA reads out as AAA, and this also codes for lysine. So when the amino acid is the same, right? When we have the same amino acid, then there's no difference, right? There's no difference here, and therefore no one is any the wiser. All you would ever know is that the sequence in the DNA is different, the RNA is different, but the phenotype would not be any different. Next, we have missense mutations. Missense mutations have two different kinds. There are conservative mutations, and there are non-conservative missense mutations. Conservative mutations, if I can go back here really quick, conservative mutations occur when you are basically changing an amino acid to something that's very similar. So if I was changing, let's say, glycine to an alanine, they're both nonpolar. They both only have one element or molecule off of the central carbon, and their structure is not very different. So we would call that conservative because yes, it's a different amino acid, but it has a very similar chemistry, it has a very similar structure, and we're not changing all that much. Non-conservative mutations, on the other hand, are when we change either the structure, so say going from an alanine to a tryptophan, or when we're changing the chemistry, so say we're going from a valine to an arginine. So we're changing the chemistry, the charge, the polarity, or the structure. So we'll go back here. Oh, sorry. So if we change polarity, chemistry, structure, any of these are non-conservative. As long as these things remain the same, but it's a different amino acid, if they're the same, it's a conservative missense mutation. So again, so we have silent, we have missense mutation, conservative or non-conservative, and then finally we have something called a nonsense mutation. This is what introduces a stop codon, or that termination sequence I just discussed, uh, on a gene, but not in the gene where it's supposed to be. Remember, you have a start and you have a stop. And so this is saying that you're introducing a stop somewhere else in the gene that isn't at the very end. So this is changing a single nucleotide to another nucleotide, and then that causes that nucleotide to become a stop codon. Now, there are only three stop codons, and so any one of those that occur before the stop codon of an actual gene is called a nonsense mutation. So there are two other kinds of mutations that can occur. These are called frameshift mutations, or what we call indels. And they are basically mutations by which um, cause a shift in the ability to read the DNA and therefore read the RNA. Now, we'll get to this when dealing with transcription translation, but effectively, every amino acid is coded for by a series of three nucleotides. On the RNA, we call this the codon because it codes for the amino acid that we're going to require. On the mRNA, I should say. So again, from the DNA to the mRNA, this has codons, which are three nucleotides big. So in effect, every gene, right? Every gene should have a number of nucleotides that is divisible by three. If it's anything more or less, so for example, normal, right? We have 9 bases. 9 bases is divisible by 3, so that is 3 amino acids. So all the previous kinds of mutations that we saw are called substitutions because we're not changing the frame, we're not changing the number of nucleic acids, we're simply swapping one thing for another. So we have GAC and we have GAA. So we're getting a substitution, not a loss or a gain. if we gain an extra amino acid, right, GAC, and then it should be T, but now we have another A. So we've gained this. Now we have 10 bases in this reading frame. That's 3.1 amino acids, and you can't have 3.1 amino acids. So we know that this is an insertion, which causes a shift in the frame. And similarly, if we need 9, if we lose a base, right, we go from eight bases. So GA, it should be an A here. Sorry, there should be a T here, but there's nothing here. And so when we've lost a nucleotide, we have less than 9 bases, or no, 8 is not evenly divisible by 3. So we know in this case we've lost a nucleotide. So in order to get at this and understand phenotypes related to genotypes, we have to understand the actual language of the DNA and the RNA in order to understand how we actually get from DNA to RNA to protein. So again, DNA, the deoxyribose, right? This has that pentose sugar that has hydrogen and hydroxide. Sorry, no, hydroxide, like so. So it's missing one of the hydroxides. Ribose, on the other hand, same thing, pentose sugar, but on the 2 and 3 um, carbons, we have hydroxide groups, right? And again, we have cytosine, guanine, adenine. And where we differ here is thymine in DNA and uracil in RNA. And, uracil being only in RNA is important because this goes back to DNA replication. You can't have uracil in DNA. So again, right, we have 2 different strands of DNA. We have one strand down here, which is the template, which is going to be the one that we're going to read. And therefore, as again, DNA, right? DNA is complementary. It is inversely complementary. So A's to T's, G's to C's. And remember, when we were replicating DNA on those RNA primers, how do we know what RNA, like what ACUs and Gs we use, is complementary to the sequence we have? So if we have an A here, we would have a U here. If we had a G, we'd have a C, right? And so we know that RNA, at least transiently, complexes, heteroduplexes with DNA. So knowing this, we know that if we're reading this, right, if we're reading this, the T on the DNA is going to lead to the RNA placing its complement, the A. The T, if we have a T, we'll get an A on the RNA. If we have a G, we'll get a C on the RNA, and so on and so forth. Because again, we're going to hydrogen bond these and then push them off as we're making our RNA. So if this bottom strand is our template, this is our coding strand. So we can just read it right across. A, T becomes U, G becomes G, A is an A, T becomes a U, and then C becomes a C. So the enzyme that reads this is RNA polymerase. And again, we're polymerizing because it's a polymerase, right? So if we're polymerizing, we're reading, right? We're reading 3' to 5', but we're polymerizing 5' to 3' because only in the 3' end do we have that extra OH, right, off of that 3' carbon. that we're going to make that phosphodiester bond. So here you go. Here's RNA. It's being complemented again. So if you have an A here, you'd have a U here. If you had a T here, you'd have an A here. And this is because this is exactly the sequence you have up here. You'd have uh, T, no, sorry, A, T, let's say G and C. So this would be C and G, because then this would be G and C. So again, every single three nucleotides is a codon, and it codes for a single amino acid. So this happens in the cytoplasm. If it's in prokaryotes, it happens in the rough ER if it's a eukaryote, because this reading, the reading of all of these codons occurs by or is done by the ribosome. And in the ribosome will, as we'll see later, has sites by which each codon is matched to a specialized kind of RNA called transfer RNA or tRNA. So the mRNA, messenger RNA, like you see here, is what sits inside the ribosome, and then tRNA gets its name because it looks like a T. And on the base of this tRNA, you have a complement of nucleotides. So if your codon is AUG, and this is your codon, the anti-codon, or the matching codons, the anti-codon, would be complementary, just like you saw with the DNA RNA heterocomplex. So this would be UAC. And UAC has charged amino acid on it that is methionine. Now, for eukaryotes, every mRNA begins with a start methionine. That's how we know that there's a start sequence. For prokaryotes, there are a couple different start sequences, but for eukaryotes, it's always methionine. And then it'll read all the way until it gets to one of those stop codons. When the stop codon comes, this is what stops the linking of the amino acids via polypeptide bonds. So if every three nucleotides it creates a codon, every three nucleotides must then dictate what amino acid it must be able to bind to. And again, these are the codon sequences found on the mRNA. And so take phenylalanine, if it's UUU, the tRNA sequence would be A, because again, it's going to be complementary to this. This also tells us that we have our three stop codons, UGA, AGA, and AGG, all of which are the three types of signal sequences to halt or stop translation. So this is another way that you can look at it. If you have one particular nucleotide and a codon, if you're starting with G's, A's, C's, or U's, what is your second codon? And what is your third codon giving you what amino acid? So again, the tRNA has a complementary three nucleotide fragment on it called the tRNA. It is charged with a specific amino acid per tRNA that exists, and it's going to be brought into the ribosome to match up with every codon in a complementary way, and this will allow the amino acids to link via peptide bonds as they process through the ribosome complex. And so we have this kind of sequence of tRNAs, which recognize codon sequences, you'll generate a peptide bond. One of the tRNAs will fall off. This reopens up the reading site, and the cycle just continuously repeats itself. So, we have specific terms for genetic information. We call that our genotype, and the resulting phenotype is a direct expressibility of that particular genotype in the form of proteins and protein products, right? So everything from height, weight, metabolism, blood type, um, the ability to even digest certain things, especially if you're a prokaryote, right? The ability to produce toxins or the ability to break down certain macromolecules, all the things that define a cell's function or characteristics are related to their phenotypes. So, the important series of terms, right? A gene is a specific piece of information that leads to a particular phenotype because either A, it produces a protein that is essential to that phenotype as the protein, or the protein does a function related to creating a phenotype in the way of, another macromolecule or a pigment or some other type of chemistry. The locus of a gene is the location on a chromosome or where in relationship to other genes a particular gene is. And an allele is any and all possible different types of a gene that that can be coded for in an organism. So, for example, like brown hair versus red hair. It's both, again, the gene or the locus is for hair color, right? But one has A and one might have a T, and that difference might be what causes the creation of the pigment of brown versus red. You could also have like sickle cell anemia, right? Sickle cell anemia both produce beta globin, but one produces a hemoglobin that has sickle cell the capability and one that is healthy, right? And so they're both beta globin. They both do a thing. They both can bind to oxygen. However, they are different. So alleles can be very similar in function. They don't have to be disease states, right? They could be something simple as hair color. They also could be disease states or could cause de novo or unpreviously seen function of a protein. Because again, there are any and all possible different combinations of genetic sequence associated with a particular locus or gene. So again, chromosome number differs in different things, right? So, for example, humans, we have 46 chromosomes. Mosquitoes have 6 chromosomes. An adder's tongue fern has over 1,000 chromosomes. And then you have bacteria that have 1 chromosome. And so again, each chromosome is going to have or I should say, for diploid organisms, right? If you have two homologous pairs of chromosomes, if you have a homologous pair of chromosomes, every locus, every gene along that chromosome is going to be the same gene. It's going to serve the same function. However, the expressibility of that function might differ because of any particular mutations or changes to that gene, i.e., the formation of alleles. So for humans, our karyotype is 23 pairs, 46 chromosomes, 22 autosomes, and one sex-defining chromosome set, each coming as haploid from haploid gametes from our parents, right? That merge and then create our own particular karyotype. So, that means that the instructions of our genotype is a mix between our parents, but again, because of meiosis and independent assortment and um, equal segregation of chromosomes and chromatids and alleles found within the chromosomes, right? And then we have somatic mutations. So we do have some traits, right? There are some cases where we have some traits that are very easy to see that are familial, but then we have other ones that are very, very different because we are all created in our own image per se. So again, when we think about allelism, allelism is a little bit more difficult to define because it's not just as simple as black hair, brown hair, blue eyes, green eyes, things of that nature. All of these are part and parcel of the ability to code for different things. And again, the function of the, and I can't stress this enough, right? The function of the gene is not per se different. In this case, the hair color gene is the gene responsible for the function of hair color. But what color is being expressed is either brown or red. And this is where the concepts of dominance and expressivity play an important role in going back to, again, how much of a certain gene are you producing, right? How much protein and where are you producing it, and what protein are you producing, and what is its function, right? And the answer to whether or not that this genetic diversity leads to very straightforward physical manifestation of phenotype is the answer is no. Because alleles can be dominant to one another, but again, recessive alleles do exist. They exist on chromosomes, they exist inside of organisms, and therefore they can be expressed. And in other cases, such as like the ABO blood type, you have different alleles that could be inherited, but they're equally expressed. And this is something that we call differences or non-complete dominance. So when we have some phenotypes which are controlled by a single locus, right? So we have again one set of genes that are responsible, and they can either be singularly expressed or doubly expressed. And in cases where you might have, say, AB blood, it's because you got one set of A and one set of B from both your parents, and A and B are equally expressed, and therefore you have AB blood. So the concepts here of dominance is one thing. So is the function or expressivity of genes from two different but important things. One of them we already discussed, which is the concept of pleiotropy, and that is how much or what kind of gene you might express that affects a multitude of different traits. So, for example, you could have um, different types of genes in the immune system, which can regulate uh, temperature of the body, can regulate heart rate, blood pressure, and activity of immune cells. It's one gene that then spans a bunch of different functions. The flip side to pleiotropy is polygenicity. So polygenicity is when multiple genes all converge to contribute to one particular trait. A good example of this is eye color. So eye color is actually controlled by something like 7 to 11 different genes. All the genes do a different thing, whether they're producing pigment or so one might be producing pigment, one might be implementing pigment, one might be transporting pigment, but they all do different things. They all therefore express and how much and how well each of these different genes work are why you get such a vast array of phenotypes related to a lot of complex genetic traits. So beyond all of this, environments can also influence genotypes and how they lead to phenotypes, mostly in the way that we discussed through epigenetics, right? We can control the expression of genes through how accessible that gene is by acetylation, methylation, chemical modifications of DNA, so on and so forth. And then we have, again, the concepts of when we have different alleles, right? Alleles can be dominant, they can be recessive, but what if you have, if we have dominant and recessive, we think of something like this, dominant and recessive. What if the dominant and recessive is a little bit more level? What if the dominant trait only expresses like so, and then the recessive trait expresses like so? Still less, but not completely. Or what if we have just two dominant traits like the AB blood type? Whenever we have two competing dominant phenotypes, we call this codominance. And then when we do have the one thing that Mendel thought he would see, but he didn't see, is when you have the blend between, say, tall and short and getting medium size, the blend between red and white. So we're blending red and white. And when we have really, truly 50-50 expression, because again, it's how much of that phenotype are you expressing, you get something called incomplete dominance, where you get that merged phenotype. So codominance is when you get both phenotypes. Merged phenotypes is when you have incomplete dominance. And again, the important kind of take home to this is when you have an allele, right? The allele is the same gene, right? It's the same gene, but the gene has a different sequence. And by and large, because that sequence is different, it has a different output. The function is still the same, color, for example, or height or whatever. But so the output is what's different, red or white, right? So red or white, because the sequence is the same. Now, there are random cases where the allele, if enough changes have occurred in a particular gene or in an allele, between two alleles, there are over time when a high number of changes occur, you could get de novo function. So that means new function. So in reality, most of the time, the allele gives you something different in characteristic, but that characteristic is still related to that particular aspect of the organism. So, for example, if we have big A, little a, right, for color, well, big A is a protein series that creates red pigment and puts in red pigment. Little a would be a series of proteins that puts in white pigment, right? So again, the function is still the same. The function is still the same, but the output is just characteristically different. So how does this relate to Mendel, right? Well, again, Mendel talks about how, laws of inheritance. What chromosomes do you have and how do they become part of the sperm and egg for sexual reproduction? All depends on which chromosome you inherit from which one. And again, every single meiosis can have something like 10 to the 13 different combinations of chromatids across the 46 chromosomes we have as humans. So between the two of them, that's somewhere around 10 to the 26 different variations. So whatever you do inherit ultimately holds all the DNA you have, including the instructions and the sequences that can be modified to be then accessed and turned into the RNA by which we can make our protein and phenotypes. Once we have created our proteins can then be furthermore subjected to chemical modification, activation, and the like. All right. So as discussed in class, please bring your questions to class when we return from break. And good luck watching this, and I'll be posting your assignments related to this shortly.